Hello champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about DNA replication. So you know there are three very important processes. One is replication, then transcription, then translation. So I thought I'll segregate and make you understand things. It will be more easy for you to capture things, yeah? So without further ado, let's just get in. Before that, what did we cover in the previous session? We spoke about DNA and RNA, we spoke about conservative, the semi-conservative method of replication, and there was an experiment to prove this. This was done by Michelson and Stahl on E. coli. So if you want the notes for the same, or if you want to see the video, you can watch the video, and if you want the notes, you can join our WhatsApp channel because it includes just us and you will get all the notes and everything there. Yeah, and I hope that your mock test also went really well. Cool. Now the number of questions which were there, children, it was 6 in the previous year, but as you can see the numbers have been much more than 6 before, so we need to prepare for that only. Yeah, and now we'll start with today's topic, that is DNA replication. What do you mean by replication? What do you mean by replication? So basics, okay. So this is a DNA strand and this is a DNA strand. These strands are attached to each other with the help of nucleotides. All the nucleotides, they have nitrogen bases which attach to themselves. So you have G and C over here, you have T and A over here, you have C and G, you have C and G again, T, A, whatever you want to do, you can do it. So these are nitrogen bases, and these nitrogen bases, basically A and T have 2 hydrogen bonds, G and C have 3 hydrogen bonds. So in this way it is attached. Now if you want to replicate this DNA, what do? You open up the strand. Okay, you open up the strand and then you will be having a new strand being formed complementary to it. So over here if it was A, then over here it will be T. Over here if it's G, then over here it will be C. Similarly the whole thing goes off here also. This strand had T, C, A. So the other strand which will be in complementary to it will be having A. If this is T, then it will be A. If this is C, then it will be G. If it is A, again it will be T. So these are the 2 new strands that are being formed, and these are the parental DNA which is already being present. Now what will happen? Basically these 2 come together and they both come together. So it might look like this. So I will make it here itself. Okay. Okay. And now this also same thing happens. Over here you have this, and over here you have the new strand being formed. So we made 2 copies out of DNA, and this process is known as DNA replication, children. Now this is not as simple as you can see over here. It has a bit amount of complexity. Okay, so we will study DNA replication in 3 steps. Okay, one is initiation, then is elongation, then is termination. We won't be going very much deep, but just at least what is there in your NCERT, we have to cover that. So initiation, as in when you want to initiate something, that is known as initiation, right? So over here, assuming that this is the DNA. Okay, and this DNA, as I said, will be having bonds over here, A, T, G, C, C, T. Okay, similarly here T, A, G, C, G, A. Okay, so if you want to replicate this, then probably the first thing that you have to do is break these bonds, right? What is the first thing that you do? You try to break these bonds which are over here, right? Now this is done by an enzyme. I'm not writing the name of enzyme, it might confuse you. So, okay. The second step that will be happening over here, first thing is breakdown. So first thing that I am doing is breaking the chain, or basically instead of using the word breaking, I can say unwinding. It is in the winded state. Now I am unwinding the DNA. This is the first step. Okay, unwinding of DNA has been done. After that, what will happen, children? Assuming that this one is 5' to 3' stand, okay, and this one is 5' to to 3 dash. You know, right? They are anti-parallel, they are opposite to each other. So if 3 is on this side, then the other has to be 5. Similarly, if 3 is on this side, other has to be 5. So there is an enzyme which helps in the process of replication, that is making a copy complementary to it. And this particular enzyme is very much famous. It is known as DNA polymerase. What do you call it? You call it DNA polymerase. But DNA polymerase, since it is the only enzyme who can do this, it's kind of very much adamant that I will be only doing my work in 5 dash to 3 dash direction. In what? 5 dash to 3 dash direction. As in, if this is 5 dash to 3 dash, it's not like it will go from here to here. No, it's saying that he will start or she will start, whatever you want to put as a gender, or basically gender neutral, whatever is there, it will be making from this end, such that this will be from 5 dash. Obviously, this has to be anti-parallel. So, it will be going from 5 dash to 3 dash direction. Cool. Okay. So, what will happen? Basically, over here if it's T, here it will be A, here if it's C, it will be G, here if it's G, it will be C, and the thing goes on without any problem. Okay. But this thing that is being formed over here, it looks something like this, where your DNA is opening up and you have another strand of DNA being coiled around everything. This particular thing, if you see like this, it looks like a fork. It looks like a fork, which is why you call this whole thing as replication fork. What do you call it? You call it as replication fork, children. So, this area where the DNA is unwinding from itself, right? It is unwinding from its normal bound state and it is forming something like this where the replication is happening. You call this region as replication fork. Cool. Now, in replication fork, this strand which is there, okay, that is from 5 dash to 3, and it's all fine because the DNA polymerase says that I will be working from 5 dash to 3 dash direction. So, it's all good. But what about them? So, basically just imagine a situation that it goes on, and this thing basically opens up. So, basically, over here, just imagine that it is opening up more and more. Okay, it will be opening up more and more just as it goes in front, because this is going in 5 dash to 3 dash direction. So, everything is going really good. But what about this strand? Basically, the replication fork is going, but you have to make things in this side towards this side. This is going this side, but here I have to make things go this side because DNA polymerase works from this direction to this direction only. And if it is 5, it is 3, then obviously here there will be opposite of 5, which should be here, and at the end you should be having 3, right? So, here 5 dash to 3 dash will be going on like this, but the replication fork is opening up on the other side. Then what do we do? Basically, what happens is that I'll explain. Okay, I'll explain what happens. So, initially when the DNA polymerase has to work, okay, it needs a primer. It needs a primer, as in, for example, if this is a replication fork, I am writing in smaller letters A, T, G, C, okay, and C, A, T, G is there. Over here you have C, A, T, G, T, A, C, C, okay, the complementary strand. Now, over here this is 5 end to 3 dash end, and this is 5 dash end to 3 dash end, okay. First thing that comes and attaches over here, which is not given in NCERT, is a primer. A primer will have exact opposite of this thing, so that the DNA polymerase gets a kickstart of things. Okay, so primer will be having A over here and then C and G over here. This is a primer, okay. From here on, our DNA polymerase start working like this. But anyway, this is going in 5 dash to 3 dash direction, just as the DNA polymerase wants. But over here, what is happening? If this is 5 dash, then this end should be 3 dash, right? From here to here, if something comes, then obviously it has to be in 3 dash, and this should be 5 dash. You see, this will be the opposite. So, end also you will get 5 dash only. But the problem is I cannot go backwards because the DNA polymerase, basically a substance, is moving forwards. Then what do I do with these strands which are being formed when the replication fork is being opened up? So, for them, there is another mechanism, that is, this process of replication is not continuous there. This process of replication is not continuous there, and for them, there will be an RNA primer over here, and then the next RNA primer over here, and then the next RNA primer over there. It goes on like that, such that every time DNA polymerase comes, it can extend from here to here, from here to here, and the list goes on. So if you see, there are fragments being made. There are fragments that is being made. This is one fragment, this is next fragment, and everything. Okay, and this fragment, what do you call it as? You call this fragment as, children, it's a very important thing, Okazaki fragment. Okazaki fragment. Okay, these fragments are known as Okazaki fragments. Now, this strand where everything is going very smoothly, okay, this strand where everything is going very smoothly, you call this strand as leading strand. Okay, because it is leading the game. It is what, children? Leading the game. So I call this strand as leading strand. Okay. On the other hand, the one where things are not going as smooth as you think it is not continuous, you call this strand as lagging strand. Okay. You call this strand as what? Lagging strand. So basically, the place where everything is going smooth, you call this as leading strand. The place where everything is not going smooth, you call it as lagging strand. Okay, and in lagging strand, you will have Okazaki fragments. So, ma'am, you said that these things that you see over here, which gives the initial kickstart for the DNA polymerase, is an RNA primer. So it will be RNA, and RNA and DNA are different. The structure is kind of different. Then what will happen? So bacha log, later what happens? This RNA primer is removed. This RNA primer that you see is removed. Okay. The DNA polymerase comes again and extends this thing, but they are not able to join the two segments. They would not be able to join the two segments. I'll tell you why later on. Okay. So every time, so for example, I removed one RNA primer, the DNA polymerase extend things, but still it is stuck. It won't be able to join both of them together. At the last point, if you see, okay, if the last point I have not joined over here, I have not joined over here. Basically, this last joining of things happens with the help of another enzyme which you call as DNA ligase. DNA ligase, children. Okay, so first thing I initiated with the help of putting RNA primers over there. RNA primers helped me for the DNA polymerase to start off its work from 5' to 3' end. But over here, even though RNA primer were there all around, okay, but since it is in the opposite direction, everyone has a break over here, and these fragments basically which are formed are known as Okazaki fragments. Okay, they are known as Okazaki fragments. Over here the RNA primer is removed and the DNA polymerase tries to complete but fails. But with the help of another enzyme is able to join these strands together. This is known as DNA ligase. So initiation has been done and then elongation has been done, and later on the whole process terminates. Now you have to understand one more thing. The replication does not just start from any point. The replication does not just start from any point, rather than that they have a specific point, and you call this specific point as origin of replication. You call this specific point as what? Origin of replication, or you also call it ORI. Okay, so if this is the origin of replication point, then from this point onwards things will be working. You have to understand that as well. So origin of replication is very much important. So on to the origin of replication, some different type of proteins comes and attaches, which are known as origin of replication complex. They all come and attach and start this process of replication, where one enzyme will be coming and opening up the whole DNA, the other enzyme will be coming, putting up the primer, the other enzyme will be coming, elongating the structure, and at the end will be terminating a lot of things. So these are it's just not one person's work, it's a teamwork. Okay, it is what? A teamwork. So what all do you need to understand? I saw one thing, that is basically in a copy I get one parental variety and I get a new strand. So since one parental variety is there, I say that this method of replication is semi-conservative. Although we discussed it earlier, I am telling you again, this method of replication is what, children? Semi-conservative, because my parental DNA is there, but not the two DNA strands are there, only one strand is there, which is why I call it semi. and it is conservative because I have the strand. Okay. Now during DNA replication, what is the most important enzyme that you need to learn? It is DNA polymerase. Okay, and DNA polymerase goes from the direction of 5' to 3' end. It goes from the direction of 5' to 3' end. If you are not able to remember what is 5' and 3', I can explain it to you, no problem. So over here, look at me very carefully. So this is, okay, a pentose sugar. Now over here, if I am marking that this is NB, that is nitrogenous base, this is second, this is third carbon, this is fourth carbon, and I have fifth carbon over here. Okay. Now fifth carbon will be bound to a phosphate group. Fifth carbon is bound to a phosphate group. Now if you go above, similarly you will have another strand, another pentose sugar like this, where you have NB and where you have C with phosphate. So basically what happens, this is 1, this is 2, this is 3, and this is 4. So here the phosphate attaches to the third carbon's OH group. Okay. So this is fifth carbon, this is third carbon. So the place where no more pentose sugar is there, at this point, don't you think that this is left like that only? It is free. So you call this as the 5' end. 5' end with the phosphate group. Okay, it's free, so that's why you call it 5' end with phosphate group. Similarly, here one nitrogen base is there, that is for example adenine. Now adenine is thymine, so another nitrogen base should be there. Okay, so nitrogen base is always attached on the first carbon. So I have to make something like this, which is the reverse of what I made over here. Okay, so over here this is first, this is second, this is third, this is fourth, and this will be fifth. Okay, so the fifth one, as I said, will be having phosphate, which will be attaching to the next one. But over here I saw the third one, which was usually attaching to this one, is now free again. Over here the third carbon, which is usually attached to the phosphate group, I told you, right, it is free now. So this end is having OH group as well. Okay, and that's why I call it 3' OH end. So if you see the whole DNA like this, so over here if there is phosphate end, over there you will be having something like this. Okay, some structure like this, where there is no one to attach to the OH group on here, 1, 2, 3, on the third carbon. So OH will be left. So this will be 3' end. So one strand will have 5' end over there and 3' end over there. Similarly, if it has 3' end over there, it will have 5' end over here. Now DNA polymerase works from this direction to that direction. That is, it makes things from 5' to 3'. Please, it makes things from 5' to 3'. It is not making, it is not depending on the strand which is going from 5' to 3' end. It's not like that. It's not coming from here to here. It is making a new strand in this direction. Okay, please understand that. Now over here, I hope this is also done. Okay, so DNA polymerase is the one that acts like that, and unwinding of the DNA is there, and this area where everything is made, the replication happens, you say that it is replication fork that is being made over there, and replication starts from only this place, that is known as origin of replication. So in prokaryotes it can be multiple, whereas in eukaryotes it can be limited, the origin of replication. Okay, it will be 1 or 2. Fine, works. Now how are these, basically when I say that one nucleotide comes, the next nucleotide comes, the next nucleotide comes, how does it basically work? You have to understand that. So what is this particular structure that I have made over here? This is adenine. Adenosine triphosphate. What is it, children? This is adenosine triphosphate. And you might be not understanding what I am trying to say. So basically, this is adenine. You might have studied A and T, right? So this is adenine. This is the nitrogen base. This is the sugar that I have. Okay, this is the sugar that I have. And over here, I have one more thing, that is the phosphate group. The structure might be, let's say something else, but as of now, just understand, just think of it as adenine triphosphate. So here is the nitrogen base. I'm just writing nitrogen base. This is basically the sugar. Okay, this is basically the sugar, and I have 3-phosphate. This is something that is important over here. Okay, I have 3-phosphate. Now, I call this whole structure as nucleoside triphosphate. Nucleoside triphosphate. What do you mean by nucleoside? When sugar is there and when nitrogen base is there, together they form what? The nucleoside. And when phosphate comes and attaches, you call it as nucleotide. So over here, the 3-phosphate, I have kept it separately. triphosphate is something separate, okay, and nucleoside is just this particular portion. Fine. So this is nucleoside triphosphate. Similarly, you have adenine triphosphate, you will have cytosine triphosphate, things like that. So many. Okay, guanine triphosphate, guanosine triphosphate, stuff like that. So basically, ATP, you say ATP is the currency molecule. Why do you say that? Why do you think that ATP is the currency molecule? Basically, what happens is that ATP, adenosine triphosphate, has 3 phosphate groups. And these bonds that you see over here, these are high energy bonds. These are what, children? They are high energy bonds which is being made over here. Now, if someone tries to break this bond, okay, if someone tries to break this bond, what will happen? This high amount of energy is being released, and this energy can be utilized for any other chemical process. I'll just repeat once more. So basically, you have 3 bonds. Now, if I break, and if there's too much amount of energy being stored in these bonds, and if I try to break them, whatever energy is stored is being released for running out certain other chemical reactions. Cool? Now, over here, what will happen? Basically what happens is that, assume there's a sugar, assume there's a nucleotide already being there, okay, and I am making a DNA from 5' to 3' end. So over here, this is the 5th carbon, right, and this 5th carbon has the phosphate group. This 5th carbon has the phosphate group. Over here there is nitrogen base, okay, this is the 1st carbon, 2nd carbon, 3rd carbon, this is 4th, and that is 5th. Now, on the 3rd carbon, I told you there is OH group, right, and the OH and the phosphate attaches, about that you know, but there are 3 phosphates, but we just want 1 phosphate. So basically what will happen, the lone pair of electrons which are there over here, they will go and attack this bond. They will go and attack this bond, which is why basically they break up. They break up. They become very furious, they become very angry. But the thing is, whatever amount of energy is being released over here because of their breakup is being utilized, okay, is being utilized for the formation of a bond over here between P and OH. So now things would be looking like this. So over here, initially what did we have? We had this with nitrogen base over here. This is 1, this is 2, this is 3, this is 4, this is 5, and you have the phosphate group, right? And over here you had the OH. Similarly, towards the down you had what? You had another sugar, you had another nitrogen base, you had 1, 2, 3, 4 and 5th carbon with 3 phosphates. You had 3 phosphate, but 2 of them left, you just have 1 phosphate left now, and you had OH over here. This OH and this, basically this phosphate comes in contact, and what do you get? What do you get, children? So this is attaching, this phosphate is attaching to the 5th carbon group over here which has OH. Similarly over here also it is attaching to the 3rd carbon group which also has OH. So it is forming ester bond. So when an acid and OH group comes together, you call it ester bond. What do you call? Ester bond. Now here also it made a bond like this, here also it made a bond like this itself. Same thing happens here, same thing happens here, you call it diester bond. You call it diester bond. Since phosphate is involved, I call it phosphodiester bond. Now who gave the energy for making this bond? Guys, these people. When they broke up, that's when the energy got released, and that's when this bond is being made. Now if it's not there, obviously you cannot be making it. So with the help of nucleoside triphosphate, children, with the help of nucleoside triphosphate, you are going to make the DNA. So DNA polymerase is using these nucleoside triphosphate. Please understand that. DNA polymerase is using these nucleoside triphosphate to form complementary strand. Complementary as in what, children? This is a complementary strand. Just like this, it is making another strand. So that is known as complementary strand. Oh, yeah. So to form a complementary strand of DNA. Okay, please remember nucleoside triphosphate. It can be adenosine triphosphate if you need A there. If you need T there will be thiosin triphosphate, or if it's G, then guanosine triphosphate. If it's C, cytosine triphosphate. But it will be in the format of this only. Okay, so that you need energy. If they break up, then only you get energy to form a diester bond, and this keeps on going. Okay, so I hope this thing you have understood. DNA replication and as well as how it is actually helping in the formation. Now one more thing, this DNA polymerase, I have different types of, there are so many types of DNA polymerases. Now this DNA polymerase is dependent on another DNA strand to form a new strand. This DNA polymerase is dependent on the information present on this strand, this DNA strand, to form another complementary strand, right? So I call this DNA polymerase as DNA dependent DNA polymerase. This is depending on another strand of DNA for forming things. So I call this DNA dependent DNA polymerase. Works? Chalo. Now we'll have a quick read of things. Fine. So DNA polymerase and deoxynucleoside triphosphate. So these things that I am seeing over here, these are nothing but deoxy. I am putting one deoxy also because in the this thing sugar, no, there is no OH. There is no OH. Deoxy, here it will be looking like just H. There is no OH. Ribose sugar has OH, whereas deoxyribose sugar does not have. Cool. Now let's see. So in living cells such as E. coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to as DNA dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. What do you mean by polymerization? Basically, when something comes together. Okay, this is one thing that is known as monomer. When something like this, so many of this comes together, what do you call them? You call them polymer. What do you call them? Polymer. And this process of making the polymer, it is known as polymerization. Okay, so that is what is said over here. They use the deoxynucleotides. These nucleotides are the polymers and they make the whole DNA. Okay, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. Obviously, you cannot expect things to be done slowly. We have to replicate quickly. The cells have to divide quickly, right? So in E. coli, which is a bacterium, E. coli, which is a bacteria, okay, over here it has 4.6 into the 10 to the power 6 base pairs. It has what? 4.6 into 10 to the power 6 base pair. How much do we have? We have 6.6 into 10 to the power 9 base pairs. In comparison to that, it is very much less, right? Now, it completes the process of replication within 18 minutes. So, within 18 minutes, okay, please have a note of this. Within 18 minutes, E. coli completes its replication and it has how many base pairs? Please remember this also. These are important piece of information. 4.6 into 10 to the power 6. 4.6 into 10 to the power 6 itself. Okay. That means the average rate of polymerization has to be approximately 2000 base per second. So each second it has to make 2000 of base pairs. Then only it can achieve the whole completion of DNA replication within 18 minutes. So this is what the average speed that should be there. Okay, what is it? 2000 base pair per second I am talking about, not per minute, it's per second, because these many number of base pairs are totally there. So you have to do it. Now, not only do these polymerases have to be fast, but they have to catalyze the reaction with high degree of accuracy. Now, if you are not doing it correctly, obviously it will be leading to what? Mutations. And mutations, as I said, it can have two effects. It can be either bad or it can be good. Most of the times we never know because our body is correcting all the things. Okay, so our body has definite mechanism, that is DNA repair mechanism. Very good DNA repair mechanism that senses things. But if by any chance the DNA repair mechanism is not able to sense things, that's when things becomes grave and we have diseases, children. Okay, so any mistake during replication would result into mutation. Furthermore, energetically, replication is a very expensive process, right? So deoxyribonucleoside triphosphate serve dual purposes. So I told you for making the DNA strand, it is very expensive because to make the phosphodiester bond that you see over here, right, you need enough amount of energy. So where do you get this energy from? You get from deoxynucleoside triphosphate. So they are saying that deoxynucleoside triphosphate have dual purpose. One purpose we know that it is providing what? Energy over here. Let's see the other purpose. So in addition to acting as substrate, so they are also acting as substrates. If they are not there, can we make the DNA strand? No. So first function of them is to be acting as a substrate. for DNA replication process. The second is to provide energy. Second will be to provide energy, children. Okay, exactly, that is given over here. So in addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in deoxynucleoside triphosphates are high energy phosphates, same as in the case of ATP. So the same thing that they have said. Okay, the two terminal phosphate as in this thing, they are high energy phosphates. Fine. Now going ahead, what did we study next? Replication fork in DNA ligase. Let's talk about it. So in addition to DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement. Okay, so for separating something, if you're joining something that requires energy, then obviously separating themselves also require energy. So you cannot expect the whole DNA to be open and then we will do the replication process. No, it happens part by part, right? So replication occurs with a small opening of DNA helix referred to as replication fork. I already told you. So this thing that you see over here, this is a replication fork. Now DNA dependent DNA polymerases catalyzes polymerization only in one direction, that is 5' to 3'. Please, at night also 3 AM, 1 AM to 5 AM, whatever time I ask you, please understand it will do polymerization only in this direction. That means the strands direction should be 3:5. So this will be a leading strand. That's when I can make something like this. So its working is from 5' to 3' end. Now this creates some additional complication at the replication fork. Consequently, one strand that is template with polarity 3:5, that is this one. Okay, the one that has 3:5, the replication is continuous. As you can see over here, there is no stop in between. So there will be continuous synthesis. So if this question is asked in which strand there will be continuous synthesis, so strand which is having 3' to 5' end, it will be having continuous synthesis because DNA polymerases works from 5' to 3' direction. Cool? Now, okay, replication is continuous, while on the other hand the template with polarity 5' to 3', it is discontinuous. It will be what? Discontinuous. This discontinuously synthesized fragments are later joined by the enzyme DNA ligase. I already told you, basically there will be fragments, and what do you call these fragments as, children? Okazaki fragments. Okazaki, not just a second. Okazaki fragments. Okay, and these Okazaki fragments are later joined by something that is known as DNA ligase. DNA ligase, ligate. It ligates something. It makes things come in contact with each other, which is why it is known as DNA ligase. Works. Now going ahead. Origin of replication. This also we studied. Let's see. So this is a picture that I have given. This is for bacteria. Bacteria have circular DNA, so they have origin of replication like this. So from here the replication will be starting. From here the replication will be starting. In the case of bacterial DNA, it goes in both the direction. But we don't need to learn that. So origin of replication is there, from where the replication actually starts. So the DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in the DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. It's not just in the E. coli DNA, it's also in us also. Okay? It's origin of replication, ORI. It is because of the requirement of origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. So, for example, I want to propagate a piece of DNA. What do? Basically, I cleave this part of DNA. Okay, I cleave this part of DNA and I insert my whatever DNA I want to replicate. I inserted whatever DNA I want to replicate. But for this, I need a vector. This whole thing that you see over here, this whole structure that you see over here, this is the vector. This is a vector's DNA. So basically, in the vector's DNA, okay, I am putting the DNA that I want because the vector has origin of replication. So then when the replication happens, it can start from here and then I can get my DNA. So that is what is said. Recombinant DNA, as in when I am making different combinations of DNA. I want a piece of DNA. If I want it to be propagated, what do? I require a vector, because a vector will be having, it provides me what? With the origin of replication. Okay, I cannot do it in my body because obviously in my body that thing will be produced. I will do it in a lab. In lab I will be using some other organisms thing, and then probably I will be making use of that particular thing's DNA for making my own DNA. Okay, works. So original replication is very much important. Now, when does replication take place? So the timing of replication. So if you are aware of things, I'll tell you. So in the cell cycle you might have studied that basically 23 hours are given for, in our case, okay, in the case of human beings, 23 hours are given for the interphase. This whole phase that you see over here, this is interphase, and the small phase that you see over here, it is known as M phase, where the cell division actually takes place. This is G1, this is S phase, and this is G2 phase. Growth 1, growth 2, and this is synthesis phase. This is what, children? Synthesis phase. Okay, so where does DNA replication happen? Basically over here the organelles, you are going to divide the cell, right? So you want to make two cells out of it. If you think you want to make two cells out of it, will it say that I will give my Golgi apparatus to this guy but I won't give it to this? No, cannot happen like this. We cannot do partiality there. So basically over there replication of the organelle is also happening. So replication of organelles happens over here, there will be so many proteins being made, things like that happening. But in the synthesis phase there is something more curious that is happening, that is DNA replication. Along with the replication of all the organelles, you need to replicate the DNA also, because it's a genetic material. So DNA replication takes place in the S phase, children, after which the M phase comes. After everything is replicated, then comes the M phase. Now just imagine that basically there is a division in cell, there is a problem with cell division after the replication has taken place. Then what will happen? This guy who needed to supposed to have one copy of DNA is having two copy of DNA because of the lack of cell division that took place. Isn't it a problem? Yes. This problem, when there will be extra amount of DNA that will be produced, you call this as polyploidy. You studied this in principles of inheritance. This is known as polyploidy. Okay, which is basically what? When the DNA is, or basically the number of chromosome is more or less. It's more. Okay, it's more. So basically what has happened, the replication of DNA, okay, we'll read from the start. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase in the cell cycle. The replication of DNA and the cell division cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy, that is a chromosomal anomaly. Okay, so the number of the chromosomes is getting messed up. So instead of having 46 chromosomes, if you have 47, it's a problem because after DNA copying, it didn't get segregated. Okay, now you will learn the detailed nature of origin and processes occurring at this site in higher classes. So the higher classes, by that you mean college or basically MBBS or whatever, wherever you are going, you will be studying extra details about it. So as of now, this is the only thing that you have to study about replication. Please remember, DNA dependent DNA polymerase. What do we need as substrate? Deoxynucleoside triphosphates we need as substrate, and they are the ones that provides energy as well. Now DNA polymerase works in 5' to 3' direction. Its direction is 5' to 3'. You have to remember that. And it does not happen, replication does not happen at any random site. It has to be happening only at the origin of replication. Works? Cool? Understandable? Yeah? Now, going ahead, what do we have? So, during DNA replication, Okazaki fragments are used to elongate what? You tell me the answer in the comment box. I will be replying to you later. Okay, please tell me the answer. I will be solving all the questions later on, but I want your answer. During replication, Okazaki fragments are used to elongate the lagging strand towards the replication fork, the leading strand away from the replication fork, the lagging strand away from the replication fork, the leading strand towards the replication fork. This came in NEET 2017. Second question. You pause and try, okay? During DNA replication, the strands separate by. During DNA replication, the strands basically separate by. Can you give a try? I don't remember if I said or not the answer, but you can definitely give it a try. So DNA polymerase, topoisomerase, unwindase or helicase or gyarase. This came previously like 1993, so we cannot expect this to come, but still you never know. Okay, so try for yourself. Third, DNA replication is conservative and discontinuous, semi-conservative and semi-discontinuous, semi-conservative and discontinuous, or conservative. What is the thing? Fourth, method of DNA replication in which two strands of DNA separate and synthesize new strand is called what? Dispersive, conservative, semi-conservative, non-conservative. Again the same question, you see different times, different ways the question has come. So you can expect anything out of it. Now last one, experimental material in the study of DNA replication has been done on. So basically if you have studied the Michelson-Stahl experiment, then only you will be able to know that. You have to tell me the answer, but we have solved it earlier, so you should probably be knowing it. Okay, now let me solve with you guys. So over here, okay, so during DNA replication, Okazaki fragments used to elongate. So you know what are Okazaki fragments and what is replication fork. So this is replication fork, children. Okay, and I am assuming that this is 5' to 3' and this is 5' to 3' over here. So basically depending on whatever things are there, A, G, C, T, C, A, G, G, whatever is there on this particular strand, there will be fragments being created by DNA polymerase from 5' to 3'. Here also from 5' to 3' end. Now this is the direction, children. Okay, these are the Okazaki fragments. So when they are saying the Okazaki fragments are used to elongate, so when they are used to elongate, this is in which strand? First thing, you have to identify the strand. Obviously if it's of Okazaki fragment, then it is lagging strand, because Okazaki fragments are breaks, right? So this has to be lagging strand. And now is it going towards the replication fork or it is going away from the replication fork? The elongation process, it's going away from the replication fork, right? So this is replication fork and it is going in this direction. So the answer will be what, children? It will be C, lagging strand away from the replication fork. Okay, during DNA replication the strands separate by, so there is an enzyme which helps in the separation of the DNA strands, and you call these enzymes as, children, helicase. What do you call it? Helicase. So this is the answer. This is an extra general knowledge that you have, but you have to know it. Fine. Now, DNA replication is semi-conservative, semi-discontinuous, conservative, discontinuous, semi-conservative, discontinuous, conservative. So, I will go with semi-conservative, semi-discontinuous, because over here, I see one is parental strand, other one is the new strand. So, I say it's semi-conservative because I am getting one from the parent, one strand from the parent. And semi-discontinuous because this side basically forms a new strand. This side basically forms a new strand continuously, whereas this side is forming discontinuously. So I cannot say continuous or discontinuous, I can rather say semi-discontinuous. So it is semi-conservative and semi-discontinuous. This came in 1989. Now 2000 question, method of DNA replication in which two strands of DNA separate and synthesize new strands is again known as semi-conservative. Okay. Now experimental material in the study of DNA replication has been Escherichia coli or E. coli, and this was the Michelson-Stahl experiment. So please remember, for DNA replication it is Michelson. Okay, there are few things that you need to remember. For search of genetic material, it started with pneumococcus. That was the Griffith experiment. After that, Avery came in during the same thing, and then came your I always forget, yeah, Hershey and Chase. You get Hershey and Chase, who did their experiment on bacterial virus. Bacterial virus as in bacteria affecting virus, which is bacteriophage. And this was the final one. this was, children, the final experiment for genetic material. It stated that DNA is genetic material. DNA is the genetic material. Now, after all this happened, we had to study about the replication of DNA. After all this happened, we had to study about the replication of DNA, as in whether it is semi-conservative or not. Semi-conservative or not. In that, it came to the knowledge that it is definitely semi-conservative, and this was done on Escherichia coli. By whom? Michelson and Stahl. Cool? So remember the name of the organisms is very important. This is the bacteria, this is again pneumonia causing bacteria, Staphylococcus, Streptococcus pneumonia. Okay? So that is all, and it's not just one question for you. I have a whole homework for you. So the next topic that we are going to do is transcription, and since we are not giving the shots now, so I want you to take a screenshot of this and then we'll go ahead. Okay? So please take a screenshot, please try to do the questions, name them 1, 2, 3, 4. So over here this is 1, this is 2, this is 3, this is 4, and this is 5. So you'll feel very free to read the portions of NCERT transcription portion and then try to solve these questions. Okay? Now, that's all, children. I hope that this particular video was really helpful for you. If you think it was helpful, then do not forget to subscribe to our channel and also comment down few things. And I hope that your Dussehra vacations went really good. So bye bye. Take care.